Good morning or good afternoon, everyone, depending on where you're joining us from, and welcome to today's webinar. My name is Freddie Lawton from Business Review, and I'll be your host. It is our pleasure to have KBI Biopharma with us today, who will be discussing complementary techniques for particle characterization. Today's guest speaker is Amber Fradkin, who is an Associate Director at the Particle Characterization Core Facility. I'd like to welcome you to our webinar platform On24. You'll notice that this webinar is browser-based, so if you disconnect for any reason, please just click on the link that you receive via email to rejoin the session. In order to ask questions, you can send them in via the questions widget, so just type them into the box at the top left-hand corner of your screen and click submit. We'll allocate around 10 to 15 minutes at the end of the session to address any questions or thoughts that you may have. If you click on the survey widget, there are a few questions in there for you to answer in order to provide feedback, and you can do this during or immediately after the webinar. Please use the yellow help widget if you require any assistance, and you can move, resize, and maximize any of the windows in front of you to get a better view of the slides. But now please allow me to welcome Amber.
Thank you, Freddie. So today I plan to talk a bit about tools available for the characterization and identification of particles, specifically with respect to pharmaceutical products. I manage, as Freddie mentioned, the Particle Characterization Core Facility for KBI. If you're not familiar with KBI, just quick, brief introduction. We are a CDMO out of North Carolina and Colorado. Lucky for me, the Particle Characterization Core Facility is actually at the Boulder, Colorado facility. Certainly, if you'd like more information about our company, you can visit our website or let me know, and I can put you in touch with the appropriate person to answer your questions. But back to the more interesting stuff, particle characterization. As I mentioned, one of our fortes is looking at particles in pharmaceutical products. And when we're talking mostly about biologics here, we have a lot of experience looking at proteins and how they degrade and how they could form non-native aggregates or oligomers that could agglomerate and eventually form particles in products. Obviously, particles come in a wide range of sizes, so we have a lot of tools to look at this from the nanoparticle range all the way up to the visible particle range. So we're talking nanoparticle below a micron up to, the subvisible range from a micron up to around 50, 70 micron, and then into that visible range as well. So looking at more of the USP 790 techniques. When we look at particles forming in this aspect, they're generally related to more inherent or intrinsic material. As I mentioned, one of the ones we are most interested in are observing the protein or active product forming particles, but certainly particles could be forming from other excipients in the material. They could be coming from your container closure, or they could be truly extrinsic material, material that you do not want in your product, such as, hair, clothing, insect parts, etc. So this is the general nomenclature we use for classifying particles within products that we look at. So where do these particles come from? If you look at, your product from the life cycle, there's a lot of factors that can actually impact particles in products. Your manufacturing process could certainly introduce particles, pumps, tubing, disposable technologies. These are all notorious for shedding of particles, as well as your configuration. You got to look at your container that you're putting your product in. For instance, you can have a syringe you expel and have one particle profile. You could expel that same syringe from an automated device and actually get a completely different particle profile. So these are all things that we help folks characterize. Your formulation and excipients, I briefly touched on that. Those can have a big impact as well on particles in your product. Certainly your shipping and handling are things you want to investigate and characterize, as well as particle detection methods could actually impact your particle profile that you're observing. One of the most obvious examples here, light obscuration versus flow imaging. You can look at the same product on both techniques and get quite different particle profiles based on those two assays just due to the sensitivities. I want to mention this because today, still, visible particle presence is still one of the top reasons for recall of parenteral products. And there's a link here to the FDA website where you can monitor a lot of the safety information. And already this year, there's been over a dozen, I'd say, recalls just related to particles. So I think we're going to take it over to Freddie to poll kind of the audience to see what folks have experience with particles in their products.
Yeah, thank you very much, Amber. Like you said, it's now time for the first poll that will be running in today's session, in fact, the only poll. So audience members, if you could please select the answer that is relevant to you and then click Submit. So the question is, have you ever had a problem, brackets, in process, bulk storage, final product, etc., due to particles? Possible answers are yes, no, and if you don't look for them, they aren't a problem. So audience members, if you could please select your answer now. And Amber, perhaps you could just briefly comment on the question to give our audience a bit more time to vote.
Yeah, and this is more of just not necessarily a recall per se, but have you ever had an issue pop up, whether it's during manufacture, during your process, or maybe a product on stability where all of a sudden you start to see particles in your material, and it made you have to stop and maybe investigate what's going on.
Okay, thank you very much. So let's have a look at the results. So overwhelmingly sort of favorite answer there with yes, that's 80.8%. What do you think, Amber? Is this more or less what you were expecting?
I was. Certainly particles are a bane of many people's job, but they've become more and more of an issue as we've gotten better at characterizing them. So probably in line with what I was guessing. The 11% saying they don't look for them is interesting too, though. Great. Okay. Well, when we discuss characterizing these particles, there's a lot of information we can actually get with a lot of the tools available. Obviously, the more established are your typical particle concentrations, trying to meet those USP limits of 788, 787, and obviously 790. But there's a lot of more information we're actually getting out of a lot of the tools available. Everything from, shape factors to the images we're collecting from these particles, the mass of particles. And probably where we focused more in the later years are actually the identity of these particles, tracking down what these particles actually are, and not necessarily just the visible range, but down in the subvisible range as well, to try to understand our products a little better. As I mentioned, we are a particle characterization lab. That's what we focus a lot on. And we have a lot of tools available to get a lot of this information for products. As I mentioned, from the nanoparticle range, we have a lot of techniques such as the SEC mols, where we're kind of looking at, the oligomers in solution, soluble oligomers. We also have tools such as the viscosizer, the nanosite, the helix, and Archimedes, which can all give us a lot of valuable information for particles in the nanometer range. Archimedes can even bridge up to the subvisible, which is a great tool to help us understand some of our microflow imaging a little better, as well as light obscuration results. And again, looking at kind of the chemical identity of particles, that's where we use our tool, the Raman microscopy, the automated Raman microscopy. Our system is the Morphology G3ID there. That can help us characterize the subvisible particles as well as the visible particles, maybe some of the extrinsic one-off single particles that are true contaminants in products. And we use these tools for a wide range of services. Obviously, we can't provide client data that's confidential, but we do have a couple of studies which I will present, which are collaborations with some of the universities or data we've collected in-house for internal use, which I'll go through today to kind of show examples of how we'll use multiple techniques to kind of characterize products. So the first example here is showing how we can use multiple techniques to evaluate biosimilarity or comparability. So this was a collaboration we did with John Carpenter out of University of Colorado, Denver, and Aaron Kruger, who has recently joined our group, so we're excited to have him on board as well. So this example, we took Neupogen and Zarcio. So Zarcio is the first biosimilar that was actually approved in the U.S. Just recently. Neupogen has been on the market for quite a while, an Amgen product there. But if we look at the briefing document provided by Sandoz, we see that they just briefly touch on the fact that Zarcio had lower subvisible particles compared to Neupogen. So obviously we take a look with MFI and we see very similar results to what they mention. Neupogen syringe here you see has very high particle counts compared to the Zarcio syringe. We had a vial sample in there for comparison as well, but mostly here we're comparing head-to-head the syringe samples. And what we also observe is that the majority of this offset is actually in the very small particle size range. So we're looking below 10 micron there, where we see where the majority of particles reside for Neupogen and Zarcio as well. So with MFI, the nice thing about that, which makes it a very useful tool compared to light obscuration, we actually get images when we characterize products. And what we see here is you can clearly see silicon oil versus non-silicon oil by images here, silicon oil being those dark, very circular particles with a little bright spot in the middle. And what we observed when we took a look at these images were that the Neupogen syringe clearly had a lot more of the silicon oil present compared to the Zarcio. So we can, there's a couple of ways you can look at MFI to try to segregate out the silicon oil versus not. There's a couple of, aspect ratio, transparencies, different morphological parameters you can use to try to understand the populations present in your sample. But where that starts to drop off actually is around 8 to 6 micron is what I observed, where the accuracy of classifying particles based on the image is actually quite difficult just due to the pixel limitations of particles of that size. So that's where we bridge that data with another technique, the resonant mass measurement. So we have the Archimedes to perform this type of analysis. If you're not familiar, this is actually a single particle detection method where we have a suspended resonating channel. So it will detect particles based on the buoyancy. So as the particles flow through this channel, the frequency with which it is resonating shifts based on that buoyant mass. And it will convert that to a mass of particle, which you can then convert to an equivalent size based on a few parameters. So what we see here when we run the samples by Archimedes, it agrees well with MFI, the MFI down there in the bottom right, that the Neupogen does indeed have higher particle concentrations compared to that Zarcio syringe. But the nice thing about Archimedes is we can tweeze this out to understand silicon oil versus not based on that buoyancy. So silicon oil will actually float as it flows through that channel versus something that, maybe protein or other material likely will sink as it flows through that channel. So we're able to understand the silicon oil population below 6 micron a lot better using this technique compared to MFI. And so what we do see are that those positively buoyant or silicon oil particles are actually the majority of the particles in that lower size range for Neupogen syringe. And if we look at the negatively buoyant particles, those are quite comparable between the materials. If we look at it, Zarcio appears to have a little more compared to Neupogen. But overall, we see that offset that we observe with MFI is due mostly to the silicon oil population. So the Archimedes will get you down to around 300 nanometers or so. When we want to look a little further, that's where we use a technology called nanosite or NTA. So this technique will actually get you down to around 30 to 50 nanometer for protein particles, caveat there. So what we're able to do is look at both concentration as well as the distribution of particles, the sizes present in the different samples. So we see again, the Neupogen does indeed have a higher particle concentration. Unfortunately, we're not able to distinguish different classifications with this technique, but overall we can evaluate the particle size distributions and see that they're quite comparable. We're seeing very similar particles between the different samples with this technique. But if we go back to those images, obviously we have a lot of great tools to help us understand the silicon oil content. But you'll observe here there's images that are clearly not silicon oil in some of these samples. So oftentimes when we see these images, one of the first questions that come to mind are, well, what are those other images? What are those particles? So that's where we use our Morphology G3ID, as I mentioned, that automated Raman microscopy. This is a really great tool. It's able to give you the same kind of information that MFI would, where we're getting images of particles and we can get a lot of shape factors out of those images with the software. But now we also have that Raman spectroscopy, so we're actually able to get after the chemical identity of these particles. And the laser's around 3 to 5 micron in size, so we're looking at single particle detection here, which has been very valuable for us to understand particle profiles in the subvisible size range. So example data here between the Neupogen and Zarsio. We see Zarsio has quite a bit of these translucent particles being identified as protein. We have a couple of signature bands, the phenylalanine, the amide, and backbone bands to pick up protein particles. We can also pick up, extrinsic materials, other inherent materials shown on the graph on the bottom right there. We see a cellulose particle, a free fatty acid particle, particles of unknown origin, which we can correlate with different Raman libraries that we have in-house, as well as purchased the BioRad libraries. But when we compare the data from Zarsio to Neupogen, we do pick up a few proteinaceous particles. We can actually pick up silicon oil if it's coated on the surface of a particle, or if we're doing solution phase, we can pick up silicon oil droplets as well. But overall, we found that the majority of the translucent type particles that we observe in Neupogen were actually related to free fatty acid, a very common degradant of polysorbate, which these formulations do actually have. So overall, we found more of the free fatty acid in Neupogen and more protein in the Zarsio for comparison. So another technique which we routinely use for folks is also another Raman spectroscopic technique, the helix. So we can combine DLS with Raman. The morphology is more of a single particle detection. This system is more of a bulk. So we're more characterizing, it's more of a biophysical type tool more than a forensic, but we can actually get information about the molecules in solution, and it is temperature controlled, so we can actually ramp that system and look at how the structure may change with temperature incubation, so thermal transitions here. Much like DSC and CD, we're kind of combining the two tools with this one instrument. So we can monitor the Raman signature coming from these two samples with incremented heating, and we can actually convert this data to a melting temperature as well as observing structural changes. And what we do see is that, these do correlate very well in how they degrade, the loss of the helical structure, an increase of that beta sheet and random coil. But we did notice that the Zarsio actually transitioned a little earlier than the Neupogen, and this correlated as well with DSC. Now, these two materials I forgot to mention are actually formulated at slightly different pHs. Zarsio at 4.4 versus Neupogen at 4.0. So this is the cause actually for that earlier onset observed in Zarsio. We did do a follow-up study which involved basically putting the Zarsio in the Neupogen formulation, and then we get agreement with both DSC and Helix with that sample. So confirming that, again, this offset was related to the pH difference. Just a quick summary here. A single method may not be sufficient to fully characterize particle profiles if you do start to see offsets when you're looking at biosimilars or if you're doing comparability studies. Careful selection of what methods you do employ, but also if you're doing this for biosimilars, a careful selection of your originator material can really help you too. A lot of times for clients, we will take material that's about to expire and literally analyze it the day before expiration to try to get a good gauge of what is normal, what's kind of the boundary of particle profiles you can get from your originator material, to then fall in line with that. So another example here. So this is showing how we apply particle techniques for actually device characterization. We do quite a bit of these in our lab. And this example is actually looking at administration techniques. So this was done with factor VIII. This image here shows the different components involved in the administration of this product. There's a reconstitution device, a catheter for an IV bolus type administration of the product, and a syringe to aid with that reconstitution. If you're not familiar with factor VIII, one of the reasons this study looked at it is there's a lot of folks on factor VIII that actually become non-responders to the material and actually have to switch different brands and try to find new therapies to help them. And one of the thoughts behind why this occurs with a lot of the patients could be aggregation present in the different materials or particles present which are causing adverse reactions in the patients. So there could be quite a bit of particles looking at those images of the different materials involved in the administration of this. So particles could certainly come from a lot of different surfaces involved in the administration. Certainly a lot of places where we could imagine either a protein could degrade during seeing these different surfaces or just the stress of the actual reconstitution administration. So this is something we wanted to characterize and mimic a type of infusion with this product. So this shows the different scenarios we looked at. We did a control there where we didn't go through the catheter. We also looked at applying different filter sizes to the end of the catheter to see how that would impact particle profiles post-administration. So we looked at a 0.2 micron filter as well as a 20 micron filter. And we also looked at the administration without a filter, the catheter with no filter. So characterizing these is really important for knowing what's coming out of your administration and going into the patient. There's just a couple examples here where studies have shown that actually adding a filter post an IV before administration to children actually significantly improved the side effects observed in the pediatric patients. So it was actually showed less adverse reactions. So this is one of the reasons why we want to help folks characterize, different infusion setups and understanding what's coming out of that and what the patient's being administered. And I'll be happy to send you those references if you're interested. Just let me know. So back to the characterization here. So if we look at the microparticles by flow imaging of the different scenarios, we see that 20 micron filter really doesn't have an impact on those subvisible particles coming out of the administration. Probably not unexpected. 20 micron is only going to remove some of the larger materials that may be present. But what we did see, the 0.2 micron filter actually significantly reduced those subvisible particles. Probably not out of the ordinary here. You would expect a lot of the larger particles to actually be filtered out with a 0.2 micron filter. So that was an interesting result. But when we look at nanoparticles, we really didn't see an impact at all with either the 0.2 or 20 micron filter. We see very little reduction of nanoparticles. So we're seeing overall billions of nanoparticles in all of these samples, with or without the filter. If we look at them with the Archimedes, the resonant mass measurement, as I mentioned, to help understand, well, are these particles silicon oil or something else? We actually see quite a bit of silicon oil in the samples. The catheter without filter here, we see almost equal populations of silicon oil as well as non-silicon oil. But what we do see with the system with the 0.2 micron filter, the bottom right graph there, we see a significant reduction of both the silicon oil as well as non-silicon oil particles present in that sample. Now, without the filter, we see a lot of particles that are non-silicon oil, so those are the red bars here. Archimedes doesn't allow us to know for sure if those are protein or some other material that is less buoyant than water, but what we can do is look at SEC mols, and what that points to is that we actually see a lot of larger soluble aggregates that elute in that void volume by light scattering, which would indicate to us that there likely is quite a bit of protein particles being measured by that Archimedes. Overall, again, one technique may not actually fully characterize your product post-administration. We do a lot of these studies, and very rarely do we just look at one single technique to help characterize particle profiles post any kind of infusion or delivery. Now, filters should not be assumed to eliminate all particles during delivery. Obviously, we have nanoparticles present that likely will not get filtered out, but we do have tools to help characterize those particles to understand could they be proteinaceous or related to silicon oil or other material. So you want to evaluate filter compatibility because we've seen some filters that could actually induce particle formation with certain products. But overall, risk mitigation strategies to help understand and potentially reduce immunogenicity of your product at the end of the day will certainly help both the patient as well as your product's success on the market. So I want to bring up too, we can certainly characterize, what would happen. We can control, a simulated infusion here in our lab, but at the end of the day, you pass those materials off to the clinic and hope that they're administered in a similar fashion. But this is just an example shown here. So John Carpenter actually works very close to a hospital, so he went over to just observe what would happen post-dilution of a product into an IV bag. And what he found was that his hospital actually had what are called tubes, where if you've ever been to a bank where you kind of put your money into a tube and then it's by air displacement, it's moved over into the bank. It's very similar to that, where it's basically shooting the IV bag several floors away to where it's going to be administered. Obviously, there's a lot of stress that can occur in this type of situation, which he did observe when they went to the other end of this tube where the bag was being basically transported. They found quite a bit of agitation and foaming, so quite a bit of stress of the product post-dilution into the IV bag. And of course, that led to very large amounts of particles as well. So this showing here a bag of IVIG diluted into the saline bag and then tubed. You see the red bars here indicating a significant increase in particle formation with this type of stress. So just something to keep in mind. We can control what we do in our lab, but it's also important to know how hospitals and clinics are handling your product as well. This showing here a lot of proteinaceous particles post-tube being formed with that stress. So now on to less scary stuff, I guess, if you will. We also do a lot of particle characterization when products are actually a particle. So an example here, this is Epiduo we purchased. It's available on the market. It's a topical, which is actually made up of 2 particular APIs. So it's a complex mixture, quite viscous, and we wanted to understand how our tools can help evaluate some of these products, because we do a lot of this work for small molecule as well. So again, we look at the morphology G3ID here. It's a great technique because it's very versatile in the types of samples we can analyze. We do a lot of dry powders. We can analyze thousands of particles to understand particle size with dry powders, as well as the chemical identity as well. We do wet suspensions. We can do solutions with a wet cell that we have available. We also have microscope slides and a couple other techniques there. We can also handle filter membranes. So if particles are not in an abundant concentration, we can actually filter that sample and collect particles on the surface of a filter for analysis and chemical identity characterization. So it's a great tool in that we can handle these gels, creams, ointments, as well as our protein solutions. And I'm going to kind of show typical lot release data here for this type of product. So generally laser diffraction is used as a lot release. So we get a nice particle size distribution with laser diffraction, but at the end of the day, we actually don't know what different APIs may make up the different pieces of that particle size distribution. I kind of think of the laser diffraction as our light obscuration with biologics. So we get data where we can look for trends, but that's about all we can do. There's not much additional information we can get out of that. So with the morphology G3ID, similar to MFI, we're starting to understand more about these products, because we can actually get images, so we can look for agglomerates of different APIs or single API agglomerates, and we can translate that back to the particle size distribution, because we can actually get the chemical identity of these single particles. So we can analyze thousands of particles, image those particles, and then go back and actually get the Raman spectroscopy. And with small molecule, we can collect that Raman signature quite quickly. So we can analyze thousands of particles in a couple hours, and it is automated. So it's kind of introduce the sample, push a button, walk away, and get your data. So this is showing here how, the lot release, the laser diffraction, we see the particle size distribution, but with morphology, we're actually able to understand what the different particles are in that particle size distribution. And as I mentioned, we can collect Raman with these particles and understand which API is responsible for each size range. So looking at this example here, the API 1 was majority less than 10 micron, the smaller size particles in the complex mixture, whereas API 2 existed predominantly in the kind of middle range, I'll say, the 10 to 50 micron. And then above 50 micron, that's where we started to see kind of mixtures where we had agglomerates of each API kind of coming together in the suspension. And so we're actually able to tweeze it out, and we can get individual particle size distributions for each API, which can help you understand, stability of these products. We've done this with a couple of products where an API can degrade into a different identity, which will actually give you different Raman signatures, so you can actually quantify how that API is degrading with time or stability or different stress, which is quite useful to help understand these products a little better. And this shows here how, it is a very robust method. It's quite useful and can be implemented as lot release in place of that laser diffraction. We get nice repeatable results with a well-developed method. And this has actually recently been accepted by the FDA, so that's created a lot of interest, certainly with our lab helping folks. So the FDA recently accepted this morphology G3ID method in lieu of a clinical endpoint. So for one of the first nasal biosimilars hitting the market in the U.S., the FDA did a lot of research into this technique and found that it was sufficient to use for characterization of this biosimilar, or generic, if you will, for small molecule talk. And there's a link there if you're interested, or I could certainly send you a link as well to read a little more about the FDA's commentary on this technique. Overall, we've seen that regulatory agencies have really shown an increased interest in subvisible and visible particles, and wanting industry to characterize those a little more, and certainly asking for things like identity when required. And we want to really emphasize that particle detection methods are certainly becoming more sensitive, so they're able to provide significantly more information. But careful management of this data is really important, and careful interpretation as well. As you can imagine, a lot of these instruments are producing very large image files, very large video files, so understanding how to securely store those data files, but also have ready access to them is really important, something people don't really realize until after they've implemented some of these techniques. But also the particle profile can be a very complex profile made up of certainly more than one identity. For instance, you can have protein particles, silicon oil particles, you could have free fatty acid particles. So understanding which component is contributing to these particle profiles can really help you understand your product and look for changes, real changes in these distributions to help understand stability. And there are a lot of methods available. So these methods differ by size and concentration, but also the amount of information they provide you. If you're ever, curious about which technique is most suitable for your product, that's certainly something we're happy to help you understand which technique could be the most valuable. But knowing your options is certainly can help you in the long run for understanding how to characterize your certain particle profile. And last but not least, I want to acknowledge a lot of the folks that help collect a lot of this data. Aaron Kruger, as I mentioned, did a lot of the work as a postdoc out of John Carpenter's lab, but has recently joined our lab, which we're really excited about. Kevin Dahl and Matt Baker are also within the particle characterization core facility, all with a lot of particle characterization experience under their belt. And a lot of these studies were, as I mentioned, done as collaborations, both with John Carpenter. We also had the device, the IV bag was done as a combination with John Carpenter's lab, as well as the University of Copenhagen with Kirsten doing a lot of that work. So I just want to acknowledge them for their efforts. And with that, I think I'll pass it over to Freddie to help handle any questions that may come in.
Yeah, thank you very much, Amber. So just a quick reminder for the audience. In order to ask questions, you can send them in by the questions widget. Just type them into the box at the top left-hand corner of your screen and click Submit. So let's have a look at the questions which have already submitted so far. Amber, I've got one for you here. Someone says, can the morphology G3ID be used to look at particle morphology in emulsion-based formulations?
Absolutely. That's one of the advantages we've seen. As I mentioned, a lot of emulsions or gels, ointments typically have relied on laser diffraction, but we actually, depending on the particle in that emulsion, we can certainly pick out the images quite nicely with the microscope.
Great, thank you very much. Another question here for you. Someone says, at what stage in development of a drug do you recommend starting particle monitoring?
I recommend as early as possible. The more data you have up front, the more it can help you as you start to progress toward that clinical and even commercial phase, especially with things like formulation development that can really help you understand stability, as well as if your formulation is the correct formulation. I know it's tough because very early on you're quite limited in the amount of material available, but some of these techniques can actually use quite small volumes. For instance, the viscosizer, you can get away with 6 microliters of solution. So as early as possible, it's really important to employ one or more of these techniques to help characterize and build that robust data set to help you assess your product, again, as you hit those later phases.
Excellent. Thank you, Amber. Got another one here for you. Someone says, in your experience, how much can be learned from monitoring of nanoscale particulates? Well,
again, a lot of it depends on what the particles are. As I mentioned, for inherent and intrinsic type particles, nanoparticle characterization can actually give you a heads up. It can really be an early warning. If you start to see formation in the nano size range, we generally see those progress and form the subvisible and even potentially visible as they reach kind of equilibrium. So for certain scenarios, characterizing and monitoring nanoparticles can be quite helpful.
Great, thank you, Amber. Got another question here. Someone says, we typically only identify visible particles, and is there a push to identify subvisible particles too?
Well, I've seen in some of my past lives, I've done a lot of particle characterization for large pharma as well, working with Amgen and Merck previously. We've certainly had questions that we had to address where regulatory agencies were asking for classification of particles at a minimum. Are these silicon oil or are these protein when you just provide concentration data? So I've seen push for identification in the subvisible size range. We've also had a lot of clients interested in identifying those just to help them understand their particle profile a little better, again, knowing, are these silicon oil, are these protein, or are these related to an excipient? Could they be free fatty acid related? In which case, something as easy as maybe a vendor switch of your excipient source could help mitigate or reduce those particle formations later. So we've seen a lot of interest in this area for sure.
Great, thanks a lot for that. Got another one here, Amber. Someone says, what's the best way to characterize particles 1 to 10 micron?
That's a tough range because you're kind of on the border of a lot of the limits of detection for different techniques. Certainly for solutions, I start with MFI that can help you understand the concentration and can help you start to classify some of those particles, between 5 and 10 micron. But then I generally suggest bridging that data between 1 and 10. As I mentioned, MFI is not very robust at classifying particles less than, say, 6 micron. So I generally recommend, if we do see an abundant population of particles in that size range, something like the Archimedes to help understand what those particles are. Certainly the morphology can go down to about 3 to 5 micron, a little lower if it's a small molecule, but it's not as high throughput as the two products two techniques I mentioned, the Archimedes and MFI. So we generally start with those two techniques.
Thank you, Amber. Next question, someone says, does one encounter different density particulates, and do different techniques account for that variability in density?
Absolutely. So Archimedes, as I mentioned, is a buoyancy-based technique. Obviously, the limitations there are that the capillary is only 6 micron in diameter, so we can't get different buoyancies for particles larger than 6 micron with this technique. So we are able to characterize positively buoyant and negatively buoyant particles. If we don't assume densities, we can certainly have that instrument output the different masses or buoyant densities of particles flowing through that system. Other than that, if we're looking at, say, density of protein particles or other larger particles, we can gauge that based on intensities, actually. So the more dense those particles are, the darker they actually appear in the image. But you got to be careful there, because a lot of those intensities will actually rely on the refractive index of the solution that protein is in. So understanding those caveats with the instrument will help you interpret that data a little better.
Excellent. Thank you very much for that. Got another question here. Someone says, from your experience, how much influence do delivery devices have on particle formation?
I've actually seen them to have a very they contribute a lot, I'll say that. As I mentioned briefly, I touched on, you can take a syringe, expel that syringe and have one particle profile, expel that syringe through an automated delivery device and have a completely different particle profile. I've seen that numerous occasions. I've actually seen where reconstitution devices, these little widgets that folks use to aid in reconstitution of, say, lyophilized products, can have a major impact on the particle profile post-reconstitution. We've seen certainly examples of that. Again, looking at filters that one may use in administration can drastically impact particle profiles. Can help you reduce those. Can help mitigate some of the particles coming out post administration, as well as the containers being used. Certainly, no container is particle-free, I will say. So looking at different bags, I think there's a couple of studies looking at different sources of saline, for instance, can have widely different amounts of pre-existing particles in them. I think maybe John Carpenter has a paper coming out soon regarding that. So they can significantly impact the particle profile. So I highly recommend folks characterize those to the best of their ability.
Okay, great, and that's about all the time we've got for this session. Don't worry if your question wasn't answered today. Amber can get back to you personally at a different time. Amber, is there anything you'd like to add before we conclude?
I would just like to thank all of those who dialed in, and certainly if there's more questions or issues you have regarding particles, please feel free to reach out to us here at the Particle Core. We love discussing particles and ways to characterize them.
Excellent. So that just leaves me to thank Amber for what was a great presentation, and to thank KPI Biopharma for sponsoring this session. To the attendees, you'll receive an email shortly telling you how you can access the on-demand version of this webinar, or you can access this through our website, which is www.business-review-webinars.com. Once this webinar ends, today's survey will appear in its place, and we would appreciate it if you answer the questions in there. And we look forward to sharing further webinars with you, so please do keep an eye out on the website just mentioned, and follow us on Twitter at BRWebinars for daily updates, and join our LinkedIn group Business Review Webinars. Thank you once again, and I hope you all have a nice day.